Hello, everyone. Thank you so much for joining us today. I'm very much excited to welcome you to our virtual classroom for a session on gene editing and how that impacts the future of research. Today, we're going to be joined by Dr. Josephine Wesley, who will be speaking to you about how scientists at the New York Stem Cell Foundation Research Institute are using gene editing in the lab to understand the mechanisms of disease, and also about an exciting tool that you may have heard of, CRISPR, that has been revolutionizing the field. The mission of the New York Stem Cell Foundation, or NYSCF, as we like to call it, is to accelerate cures and treatments for the major diseases of our time through stem cell research. So we're very much thrilled to have you join us today to learn about one way we are striving to achieve this mission. A quick housekeeping note, questions can be put in the Q&A, or at the end of the session, you can raise your hand and we will be able to unmute you. So thanks again for joining. And without further ado, let me introduce Dr. Wesley. Josie, take it away.
Thank you, Richard. Hello, everyone. Good afternoon. I'm looking forward for the next 40 minutes to introduce you to gene editing and research and translational medicine and go through that journey with you, and hopefully you all can learn a little bit from me and we have a good discussion at the end. So my name is Josie. I want to go a bit more into the personal side at the beginning, and I'm a scientist, and I think before we start with gene editing and CRISPR Cas9 and all these things, with the last, few years, the question of why did you become a scientist became more and more. I got this question more and more often. So I think I would just, tell you a little bit, like 1 minute, about myself. So I was diagnosed with type 1 diabetes when I was a kid, around 10 years old. And I think that probably triggered my curiosity and my need to figure out more about my own disease. And maybe a lot of you or some of you have a family member or someone in their surrounding that also has some kind of trouble. It doesn't need to be, something like type 1 diabetes, an allergy, anything about that. And you start questioning, why does that come? And so a lot of people in my environment, which are also scientists, have exactly the same background and were super interested in biology, have family members or, friends that have some kind of disease, or, as I said, even an allergy counts for that. You become very curious about why do I have this disease, or why does my friend is a bit different than me? I also figured out for myself that I like optimizing, and I was never satisfied with how my type 1 diabetes was treated. So I was like, that needs to be better. So that's, a few things that really makes up a full scientist. And if you feel yourself, in a very curiosity, that you're a very curiosity person or very much interested in biology, you might consider going for that without making an advertisement. But so let's dive into our topic today. So what I want to tell you a bit is about how scientists use genome editing. I want you to understand why and how DNA damages can even occur naturally. So, when we talk about genome editing, a lot of people right away think someone else, a scientist, is changing something, but DNA can change basically naturally. I want to dive a bit into then the CRISPR Cas9 genome editing that probably most of you have heard, and that's why maybe you're interested to be here. And then one of the biggest points, or very, very interesting discussion point, is obviously, what do we know about the advantages and also disadvantages or pitfalls of genome editing, and how do we need to discuss that in the future when applying that? So I wanted to make that a bit interactive, and so I will ask you during the webinar a few questions. And so here you will see the first poll, and it's about how do we get from DNA to an organism? And I'm pretty sure most of you have an idea how that works. So when we start with DNA, what would be the next level we go for, and how do we end up with an organism? And I basically want you to select what you think is the right answer. Let's see. Okay, great. I see most of you got it right. So the answer, I don't know if you can see it, but I will show you, is yes. We go from DNA to RNA. We make an amino acid change, which will be folded to protein, which a lot of proteins make up cells, and then we can build an organism. And that's important to know, because that's where we start, right? We want to interfere with the DNA, and at the end, something will happen in the cell or an organism. So I want to make sure that we're all on the same page. So what is DNA, right? It's deoxynucleic acid, and it consists of thousands of billions of small units that we call nucleotides or bases. It forms a helix, like a twisted ladder-like structure, and it is in all our cells. Each of our cells in the nucleus has DNA. And it is basically the genetic code and really the basic and most important information that determines how we look, or how healthy we are, what is our eye color, what is our hair like, and a lot of more, right? And so, as most of you got it right, we go from DNA to RNA, amino acid change. These needs to be folded to proteins. It will form cells, and finally an organism. And I want to go a bit deeper into the DNA, RNA, amino acid, because it's super interesting. It is interesting for the reason that the DNA basically needs to be encoded. You know, it's kind of a Sherlock Holmes strategy here. So you know the DNA is a double-strand DNA, and it has all these bases like A, T, C, and G, and then it will be transcribed into RNA. But then we go from RNA to an amino acid change, which completely looks different, right? I mean, the DNA and the RNA look fairly similar. We know that the T will become a uracil in RNA. But other than that, how does that go into a protein chain? And scientists, looked into it and figured out there is actually, it's a code. It's, a genetic code. And most of you might have seen this kind of, we call it codon, or codon. And when you go in your DNA from the left side to the right side, you can go from the inside of the codon to the outside. So let's take as example the last one, which is the threonine, right? It's an ACC 3-letter code in the DNA. It will become still an ACC in the RNA, and then it will be translated to a threonine. So if we follow the A in the middle of the circle to the next C, and then to the next C, you will find threonine in here. And what you also see, that threonine doesn't have only this 3-letter code ACC, it can also be ACG, ACA, or ACU. And so it is, in the end, if you figured it out, not that difficult. And therefore, I want you to figure out what is the alternative third base for glutamine. So if we look at the beginning, we have GAG. And if you go and use the codon from the inside to the outside, you will figure out that there is one alternative base for the third position. And I want you to tell me which one is that. Okay, I guess it was not that easy, but most of you got it right. And I will show you the result here. So again, you go GHE from the left to the right, and you go from the inside of the codon to the outside. So you go G, A, and G. And the second letter that's in here as an alternative for the third letter is an A. So that's kind of, I know it was not that easy, and it's kind of tricky, and it needed some time for scientists to figure that out. But now it helps us actually to understand how the DNA becomes at the end a protein chain, folds protein, and finally we have cells and cell material that can be used in there. So there were a few basics, and so now I want to really dive into the gene editing part. So what is so exciting, or why is this such a big topic in the scientific field? If you look at your Zoom window and you just look at your neighbor, the differences between you and this neighbor in the genome is like millions of bases that are different. And these differences can interact with each other, and therefore you either have different eye color or different hair, or if you're unfortunate, you might have a disease, which doesn't always need to be severe, but sometimes it is severe. And for us scientists, diseases are very interesting, and obviously we want to investigate that much more, and how does it come that the genome is connected to these diseases. And so gene editing finally allows us to understand which of the genes are important for specific diseases, and it basically really helps us to move forward in the field. And with CRISPR technology, and I will mention a few other technologies that we used in the past for gene editing, we really got a very cheap and fast way to modify the genome in cells, which obviously again accelerates our knowledge and our knowledge of diseases. So when we talk about changing the genome in cells, there are two categories that I really want to make sure that you all know the difference and distinguish between them. So we have somatic cells and germline cells. And so we can do gene editing in somatic cells, which would only it would stay either in this person or in this kind of cell type. It would not be passed down to the offspring. So there are approaches where, people donate blood, and the blood cells will be gene edited and can be reintroduced into the patient. But these cells will only stay in this person and in this patient. And if there is an offspring, like if you have kids, it will never pass along. But then there are the germline cells, and there is also, obviously discussion about how we could edit the genes in the germline cells, and that would actually affect your offspring and the next generation and the generation afterwards. And both of these things have, considerations where you might think, oh, we should do it or we should not do it. And it's a big discussion in the field. And I also want to, give you all the opportunity to think about it and discuss about it a bit. And so that comes to basically my next question. And what do you think, why someone would like to change the sequence or someone else's DNA? And I gave you, and I will read them for you, I gave you some options, and these are not all the options that we have. And there's also don't be scared, there is no right or wrong. I just want to get an idea what you think. So there is cell replacement therapy with gene-corrected cells, to cure disease. You could enhance the human abilities when you do germline editing, or even simply change eye color or hair color or height of a person. You could just prove it because, I'm able to do it, so I want to do it. You could gene edit an embryo to prevent maybe a life-threatening disease if you know it beforehand. Obviously, you could also just do it for research purposes and try to understand diseases or try to understand genetic influences And one aspect is also, you can not only go for human cells, you can also change plant cells or other cells, for example, for the food supply. So I would like to know, what is your opinion? What would you say is maybe for you right now most important or most reasonable? Okay, I see. So most of you, I hope you see that, say it would be cell replacement therapy to maybe cure a disease. There are a few other options. And as I said, there is no right or wrong. It's more like I want to keep the discussion. I want to see what you are thinking. And so my personal guideline, or my personal hierarchy in that is shown here. I would, since I'm a scientist and I really see that this is a big advantage for research purposes, there is no big doubt for me that this is not a good idea for research. I also again, it's my personal view. There is no right or wrong. I think improvement of plants for food supply, since we are a growing population on the world, would be something. And then it goes down the line. I feel like nobody should do something because I'm just able to do it without, a good reason and a good background. But as I said, there is no solid line and there is no yes or no. It's just, I want to give you some thoughts about it and keep the discussion going. And so for the next few slides, I really want to dive a bit into gene editing. I first want to tell you what I'm doing here at JAX-NYSCF with my team. And I'm actually leading a team that's called Functional Genomics. And we don't only do genome editing, we also do disease modeling. And that's where things come together. So it's really, really interesting. As I mentioned before, just imagine there is one gene that maybe we identified has two variants. Let's not call it a mutation, let's call it a variant, because we're all different and maybe a variant is not always bad. And so let's say this gene has one time an A at this position and one time a T. And so in my team, we would differentiate cells with these genomic information into liver cells. And what this means is that this gene one time would produce a protein that is very important for fatty acid metabolism. And the second one would maybe make a protein that's not properly functional. And if we look at liver cells, maybe in the first case, we have, one of the important things for liver cells is to remove fatty acids. This would work pretty well. But in the second case, with this gene variant, it would not work very well. And this is really where gene editing and disease modeling are coming together. And it's really super interesting and exciting. How else we can investigate this kind of genetic information and when it comes to the phenotype. So what we could do is obviously try to repair that variant or change this variant to the other variant and see if the phenotype, so how the cells behave, becomes the same as the original one. We could also take the upper variant and just knock it out, delete it, and see what happens to the protein and what happens to the cells at the end. So there are different approaches that you can use gene editing to answer a very similar question. And sometimes you actually need to do all these things to make sure that you have the right answer. If you look at JAX-NYSCF in general, there is much more that we are doing. And the functional genomics team and my team is just a small subset in the whole picture and the whole story. So what we do is usually we reprogram blood cells or fibroblasts from patients or donors into induced pluripotent stem cells. So as you know, we are a stem cell institution. And stem cells have a really great ability to differentiate in any other cell type. So here is the disease modeling part coming into place. And so my team is doing the gene editing on induced pluripotent stem cells. And then we look at the edited lines versus the non-edited lines and differentiate them into the cell types that we want to differentiate. And I will give you a few examples for diseases. It is it is tough because even myself a few years ago would not know all these diseases. But we work on a lot of very rare diseases. One of them is the Alexander disease. So there is a mutation in the GFAP protein. If I really summarize in very, very short, it's about the myelination of neurons. And there is a defect in this myelination. And one of the advantages working with induced pluripotent stem cells is that this is really, really hard to model in animal models. So as a researcher, we need to, go for the stem cells and differentiate them into the cell type that we want. And with the gene editing now, we can repair this mutation and look if we can actually reverse this phenotype. There are a few other diseases. If you want to look them up at some point, it's really interesting, and they're very rare and specific. One is the infantile neuroaxonal dystrophy, Batten disease, and Jordan's disease. But JAX-NYSCF is working also on a lot of common diseases, and probably everybody of you heard about Alzheimer's disease. And what you can see here in the graph is that one of the important parts when you do stem cell research, and especially disease modeling, is that you're in the right cell type. So here you see a graph with different variants of the APP gene. And you see in the iPSC lines, there is no real difference between these lines. But since Alzheimer's disease, is a disease of the brain and the brain cells and neurons, the moment you differentiate them into the cell type that is usually affected in people, you see the differences. And that's also super important. And so Alzheimer's disease is one of the diseases, and there are a lot of variation and different forms of Alzheimer's disease that we're working on. Parkinson's disease is another one, and diabetes is one of the diseases we're also working on. And so coming back to the genome editing, I want to make you all aware that CRISPR Cas9 is not the first technology that we are able to use for genome editing. Before that, and I even worked with TALEN in the past, there were two other basically technology or enzymes that were used. So TALEN is a transcription activator like factor nuclease, which they all do the same. They all are able to specifically find the DNA sequence and cut it. The second one is the zinc finger nuclease, which is a more artificial enzyme that was generated to, again, do exactly the same, doing DNA cleavage. But these two technologies are a bit tricky, and they're not so easy to apply and to use. So that's why we're now mostly using CRISPR Cas9, because it's really, as I said before, cheap and fast, and it's not so difficult. So I will show you this small video and explain what happens. So the big round thing is the CRISPR Cas9 enzyme. And what you need is the red part here, which is a guide RNA that is complementary to your double-stranded DNA sequence. And Cas9 is basically using this guide RNA to find the sequence that you want to find. And it cuts the DNA, both of the strands of the DNA, into two. And then the cell has to deal with a double strand break. And I'm coming to that point in a second. So going back to the CRISPR Cas9 again, because the movie might be a bit fast, but so the big cloud here is the Cas9 protein. We have the guide RNA, which basically anchors into the Cas9 enzyme. And then in green here, it has this complementary sequence to your double-stranded DNA. And one of the really, really important parts to have with Cas9 is the protospacer adjacent motif called PAM. So the PAM site needs to be next to the complementary sequence for the guide RNA. And why is that? It's basically a start button for the Cas9 that says, okay, you're in the right position and you have the right signal, you can cut, you can introduce a double strand break. And so there are different CRISPR caspases. So different versions of this enzyme have different PAM sites, but the Cas9, which is the most common one, has a PAM site that's NGG. And N in this case stands for any nucleotide. But then it needs to follow by 2 Gs. And so my next question for every one of you is, you see the sequence below. And it's a long sequence. It's going from 3' end to 5' end. So I want you to look for as many NGGs as you can find, and tell me how many you think they are. I know it's not that easy. A lot of letters. Okay. I don't know if the poll is over, but I will give you the answer. I haven't seen it. But so here's the answer. There are 4 NGG sequences. I know it is tough, but this is basically the first thing that if you are planning and designing a gene editing experiment, you need to know if there are these PAM sites. Otherwise, your Cas9 cannot even cut. And nowadays, we have, softwares and bioinformatics that actually can screen through the whole DNA sequence to help us with that. Otherwise, project would not move fast. But that's pretty cool. But so what I just told you is that the Cas9 is basically breaking our DNA. And I just want you to be sure and to be aware of that our DNA is breaking, or DNA changes are occurring all the time in our body. And that happens, on a natural or unnatural way, but you all know UV light, for example, is not good for your cells, and it introduces actually double-strand breaks. Even by just replicating, if your cells just replicate, sometimes, mistakes are happening and there is kind of a stress for the cells. So it can even happen during DNA replication. And so one of the big questions is, what happens after the DNA is broken? It doesn't matter if the Cas9 did it or it happened because you were too long in the sun. What happens in your cell? And it is important to know that, because if you just introduce double-strand breaks into your cells, there is no way, how would you fix it? How would you do targeted gene editing? And so the cell is capable of a lot of things, and one of them is to repair their own DNA. And so I simplified that here a lot, but basically what happens, you see, we have a DNA, we have a double-strand break, and then there are 2 pathways that can happen. One is without a template, and your cell basically will either remove a few nucleotides or add a few nucleotides at the breaking point and stitch it together. And that usually results in small insertions or small deletions of your DNA. And a lot of time, if that happens in your cells, it happens in one of the regions where there is no gene, or at least we don't know that there is a really relevant gene. So it is not a big problem. But if the cell has a template, and sometimes it's going for the other pathway, it basically can use this template to repair the DNA strand exactly in a way that the template looks like. And as you all know, we have a chromosomal set of 2, so every gene exists 2 times in your body. So you actually have a template in your cell and in your nucleus itself. As a person of gene editing, we try to introduce this template. So we will give to the cells the Cas9, which breaks the DNA, but we also will give a lot of template to use this template exactly in a way that we want the cell to change. So example, we could change the mutation into a wild type, or we introduce something that was not there before. So we don't only need the CRISPR Cas9 for gene editing, you also need to a way to repair your DNA. And so now you know a bit more about Cas9 and what it's doing and what can be, what can happen and what the cells need to do actually to overcome this double strand break and repair it again. And I quickly want to touch base on things that actually can go wrong. Like we call them off-target and on-target effects. So off-target effects are when the Cas9 goes somewhere where it's not supposed to go. So let's say the guide RNA that you used finds another area in your DNA which is super similar to the one that you actually want to target, and it can just introduce a double strand break there. And you are maybe not sure if this is an important area or not. So that's what we call off-target effects. There are also on-target effects, and that's more like if you target a specific gene and something happens that you didn't want there. So maybe you wanted to repair it, but it didn't work out the way you wanted it, and you introduced some kind of an insertion or a deletion, and the repair didn't work out properly. And it is because it is on the target you wanted, but it's something that you actually didn't want. It's an on-target effect. But in general, I have to say, if it works, it can be very precise, and it can be really helpful to understand a genetic mutation and to actually, understand why this gene is important or why this mutation is leading to a disease. There are also ways to make sure that your cells are okay. So you can identify potential off-target sites. Again, bioinformatics is really a big topic in that and will help a lot. And then you can basically check on the sites if the Cas9 targeted or cut there or not. And in the end, as I said, you can use gene editing for sure in research, and you can overcome all these disadvantages and make sure you have good controls and your experiment is set up in a very good way. And so I'm almost coming to the end. And so after I told you all of that, right, we talked about what we can do, what cell types we have, the somatic and the germline cells, while the germline cells will be passed, or the information on these germline cells will be passed to the next generation and the generation afterwards. But then we also have the somatic cells, which stay exactly in the body of the person and will not be passed. We talked about double strand breaks, and they can also occur naturally, and that Cas9 basically the main job of the Cas9 is to introduce the double strand break, but hopefully very specifically. And then we talked about how the cell itself needs to repair the double strand breaks and how that can go wrong or not. And so again, I just want to point out, when we discussed in the beginning, or when I asked you in the beginning, what do you think? And where should we draw the line? What is important? What can we do? What are we allowed to do? It is an always changing field, and the more knowledge we have, the more we might change our opinion on one on the other. So I just wanted to bring that back up. And so for the last 5 or 10 minutes, let me check the time. I think we're good. I want to give you the opportunity to ask some questions, and obviously ask me as many questions as you want. I will try to answer them all. I put some of you, some of the potential questions already in here. So you can ask me something about the background or the method. I'm doing that on a daily basis, so I should know most of those things. Ask me or discuss about advantages or disadvantages. Maybe you changed your opinion throughout the presentation. Maybe you didn't for a good reason. I would be very curious to hear what would be the reason if you changed your opinion or not. again, what would you use it for? Do you have any idea, like what would be your thing? Or maybe I have a friend who has a disease, I would like to use it for, helping him, or I would like to investigate in something that a relative is suffering from. And the last question, obviously, you can also ask me just about how I became a scientist. Also, I told you a bit about that. But about me, what I think personally about all these things, and any kind of question is very welcome. I don't know if I go to step by step, or I just give it to Richard, and he gives me a few questions.
Thank you, Josie. That was an enlightening presentation. I like your use of Spider-Man, Spider-Men, besides the point. As I mentioned briefly before, please feel free, I already see some questions coming in the Q&A. Please feel free to ask away in there. If you feel brave enough, you can also raise your hand and we can unmute you to ask Josie directly.
Should I keep the screen, or should I stop sharing the screen, or it doesn't matter?
I think you can keep it up.
Okay.
Just to keep it going. I have a very specific question here from Samantha. Are we able to fix epigenetic changes with CRISPR?
That's a good question. So there are, like, CRISPR technology is now used for a lot of things, and I guess you heard about epigenetic modifications done by CRISPR. So we are trying, and for now it's basic research, and we are applying CRISPR to do that. I have to say I'm not the expert in the epigenetic field, but for sure people are using CRISPR for epigenetic modification. I'm not sure how well it works, how long lasting it is, because obviously epigenetic can change when the cells are changing, when the environment is changing. While if you do a gene edited, and if you do an edit in your DNA, and it was successful, it's very unlikely that it will be reversed. While, epigenetics is very flexible, and factors can come back and change the epigenetic status of your cells. But yes, it is used, and we are moving forward, and it's a really good question.
Thank you. Next, from just to keep it within the scientific realm, I'll ask you about your career soon. From James, in a human, how do you get the CRISPR machinery to cross the blood-brain barrier? Do you have an answer for that? And especially if you want to target a specific region of the brain.
That's a good question, and I don't have a clue, to be honest, if we can do that. What we are doing, as another example, to edit, one of the best examples is probably blood cells, and it's the most accessible cells that we have, because you just need to draw blood. And what's happening is actually that people, that have, maybe you heard about sickle cell anemia, they get their hematopoietic stem cells, their blood stem cells removed, gene edited, corrected, and reintroduced in their body. To be honest, I am not the expert in the blood-brain barrier, and I would not know how to bring CRISPR in there, because even compounds and drugs, are difficult to apply to brain diseases.
Right. How would you say gene editing CRISPR Cas9? You mentioned it a little bit before with Alzheimer's, but how would it be used in Alzheimer's and Parkinson's disease and neurodegenerative?
Yeah, so for now, we're doing a lot. For now, Alzheimer's disease and in general, all these age-related brain diseases, I believe we still don't know enough about it. And so mainly right now, CRISPR is used to identify the genes that are responsible for it. And we are more and more coming to the conclusion that there is not only one gene, which makes the whole, story even more complicated. There might be several genes that are, in context together. There are risk factors and non-risk factors. So in order, I mean, for now, for the brain diseases or Alzheimer's disease, it's more a tool to figure out which of the genes, might be responsible. And then if we know, and if we can show and prove that this gene is responsible, then we can find a target, meaning a compound or a drug or a medication for it. Yeah.
Yes, it seems we're in the earlier stages, but it's very, very promising.
Yeah, I think we have never been like with CRISPR-Cas, the knowledge and the understanding of Alzheimer's disease and this, brain diseases accelerated a lot. So I understand that a lot of people are like, how can we use it to cure someone right away? But just imagine the amount of more knowledge we have and more potential targets that we identified with this technology to maybe get to the point to have something that helps Alzheimer's patient is already really promising.
And it seems to be also just a growing field in general,
Exactly.
which we can shift gears a little bit and then ask about you, Josie. Maybe a two-part question. How exactly did you get into this field of study? And maybe afterwards you can answer, what is the level of education that you suggest for someone who wants to do this type of research in a role like yours?
Yeah, so as I said before, I was diagnosed with type 1 diabetes and I started reading publications. And I started getting very interested in biotechnology. And so I decided to study biotechnology. And honestly, I never wanted to work on my own disease, because if you start working in science, you also realize that a lot of things go maybe not as fast as you want them to go. So in the beginning, I was like, I really want to do research and disease modeling, and I want to help other people. And I hope that someone else is focusing on my disease, because it might get frustrating. But yeah, that's how, I mean, really being affected by something that you really cannot change, but you maybe want at least to understand it and move the field forward, was a really big trigger for me. And therefore I went for biotechnology in my bachelor and also biotechnology in my master. And then I did a PhD. There is no, for the career itself, there is no straightforward answer. And it's also the scientific field. I mean, science, like a lot of things are science, right? You can go for bioinformatics, you can go for machine learning, you can go for biology, neuroscience. And I think the first step is to find something that you're attracted, that motivates you to do a bachelor in this field and maybe do an internship or something. But it is super important to do a lot of trainings and internships to figure out which direction you really want to go. There is nothing worse that you end up in a field that you focused for 10 years on, and you realized it's actually not what was your interest in the first place in science, because science is a lot of self-motivation and a lot of focus and thriving, and it is a tough business. But like, don't plan, if you're now at high school, don't plan to go for a PhD in 5 years. Do it step by step, right? So what is my interest? It's maybe neuroscience. Let's do a bachelor. Let's go to do an internship in neuroscience. Figure out if you really want that step by step. Yeah, I guess that's my answer.
Thank you for that. Just the interest of time, I think we're going to have one more question. What do you find most hopeful about the field of genome editing?
Most hopeful is that, so for me, most impressive, and that gives a lot of hope, is that originally, years ago or a few years ago, when CRISPR-Cas was identified, someone said, like the scientists who identified said, oh, we can do genome editing. But as the first question actually implied, it can do much more, right? We can try to change the epigenetic. It is not only used as genome editing, it can be used as an enhancer, an inhibitor. So you can actually modify this tool in a way that it doesn't cut your DNA anymore, but basically enhances the expression of a gene. So you don't necessarily need to modify. And so the fact that, and this will come again and again with a new tool and another tool, the fact that someone brings up one tool and so many different scientists come together and use the tool for actually something, you know, to work on a little bit something else is for me impressive. And it tells me that we are moving really fast into the right direction. And it gives a lot of hope. Also that, I mean, going back to the question actually, sorry. I mean, first of all, it's impressive. It's just impressive how technology, you know, one protein is used for different things. But then, as you all know, it's here for about 10 years. And we have actually clinical trials. Like what I'm saying, that everything accelerated and the knowledge accelerated with CRISPR-Cas, and we can use it in a very smart way, also points towards there are actually clinical trials and there are actually therapies now, for example, for sickle cell anemia, that apply CRISPR-Cas, and it's safe enough, you know, for these people, and they can have a better life than with the sickle cell anemia. And so as a type 1 diabetic person, I'm waiting for my cell transplantation in the future. Yeah, it is super great to see how everything is like moving really fast in the right direction.
Yeah, definitely. I mean, during COVID,
Exactly.
We're seeing not only technology and advancements, but also collaboration and innovation. It's truly an exciting time.
Yeah.
Well, thank you so much to everyone that joined us today. And thank you so much to Dr. Wesley for your presentation and for answering our questions today. Please take a moment to answer our survey that's just been put in the chat. Tell us what you think about our presentation today. But thank you all so much again, and happy holidays.
Thank you.
Bye, everyone.